Outstanding. Thank you again. So no better place to talk about the future of clinical trials than at Christian's conference when they think about the future of digital biomarkers. So I'm delighted to be here again and just admire the work that Christian and his team have done in terms of developing a wide range of digital biomarkers, especially for neurological conditions, as I'm a neurologist. So I'm going to talk a little bit about the rationale for the changes that we're going to see in clinical trials, and I already alluded to them earlier. And I think talk about two changes that are going to occur, and I think they're married to one another. One is decentralized studies that bring researchers, clinical investigators directly to participants. And the second is using digital biomarkers to get real-world objective assessments of people in their natural environment. Neurological diseases are the leading source of disability in the world, more than infectious diseases, including COVID, and more than cardiovascular disease, and more than cancer. And many of these conditions are growing rapidly. So I care for people with Parkinson's disease, and Parkinson's disease is the fastest growing neurological disorder in the world. Over the last 25 years, the number of people with the condition has more than doubled. And in the coming 25, it'll double again if we don't change. Yet the way we've been approaching drug development for these neurological conditions has largely been unsuccessful, maybe with the notable exception of MS, where we actually have an objective measure of the disease, but where we don't have objective measures of the disease, where we rely on rating scales to determine whether our drug works or not, whether that's in Parkinson's disease or Huntington's disease or Alzheimer's disease or ALS, we fail, and we continue to fail. And so this is just a list of a number of drugs for Alzheimer's, ALS, Huntington's, and Parkinson's disease that all thought they saw a signal of benefit on the subjective rating scales in phase 2, but when they went to phase 3, there was none. And so solanezumab for Alzheimer's disease, they thought they saw a 1.9 point improvement on the ADAS-Cog in phase 2, but when they went to phase 3, they saw none. In some cases, it was so clear that they saw none that the trials were terminated early for futility. And these failures cost thousands of people years of their life, and cost pharmaceutical companies hundreds of millions of dollars, and cost society a lot of suffering. And so what do these rating scales look like? So this is state of the art for measuring Parkinson's disease in 2020. And so I'm going to have you guys all do it at home. And so we're going to take our thumb and index finger as big and wide as you can. And I want you to tap it big and fast 10 times. And then based on how well you tapped your thumb and index finger 10 times, someone like me rates that as 0, no problems, 1, slightly impaired. The regular rhythm is broken by 1 or 2 interruptions. There's slight slowing, or the amplitude decreases toward the end of the 10 taps. Mild, 3 to 5 interruptions. Moderate, more than 5 interruptions. Or severe, you can't even do it. So you can see there are a number of flaws in the way we measure it. One, it's subjective. What I say is a 2, my colleague might say is a 1, and another colleague might say is a 3. All that variability biases you toward the null, biases you towards finding that there's no effect of the intervention when there may be. Second, if you have Parkinson's disease and you're participating in a clinical trial, you're either 1, 2, or 3. So there's not much room to change. So you can maybe go from a 2 to a 1 or a 3 to a 2, but that's it. It's a categorical scale. And this rating scale has not been designed to be administered remotely. So you need to be in person in clinic. Yet we know people who are older, people with Parkinson's disease and ALS, for example, are at higher risk for complications of COVID. So in short, we have instruments that could be less well suited, could be worse suited for our present environment than they do now. And I picked on Parkinson's disease, but the rating scales for Alzheimer's disease are even worse. So this is actually a good rating scale as far as rating scales go. So I want to talk to you a little bit about decentralized studies. And so Andy Corvos, who's outstanding, Christian, you should invite her to speak next year if you haven't had her speak. She's co-founder and CEO of Electra Labs, and she wrote this really great piece on decentralized studies, and she looked at 2 dimensions. She defines decentralized studies as having 2 dimensions. Where are the data captured? Are they captured on the y-axis? Are they captured in the clinical site, or are they captured in the patient's home? And then how are the data captured? Are they captured via an intermediary like a rater like me, or are they captured via a smartphone device like you've developed for MS or for Parkinson's disease? And that traditional clinical research has data captured at the research site, so filling out a rating scale, and it's done via an intermediary, someone like me. An entirely virtual study would use a smartphone to capture data, and that data would be captured in somebody's home environment. And you can imagine you can have a wide range of studies that might be hybrid in terms of how the data are captured, or hybrid or where the data are captured, but entirely virtual for different aspects of it. And these virtual models or decentralized models have some significant advantages. One, a fully virtual model, recruitment is determined by internet access and not by site location. So if you do an entirely remote clinical trial, you can do it broadly throughout the United States or even globally throughout the world with a small set of investigators or a few set of sites, fewer institutional review boards, shorter time to initiate the study, shorter time for visits, decreased variance in assessments, much decreased burden on research participants. Think about if you had ALS, what would be your idea of like the least palatable thing to do is to hop into a car and drive to a major medical center to be potentially exposed to infectious diseases even before COVID was around. And we think that the cost for these virtual studies can be less than the traditional model, which is very, very time and resource intense. And it's only going to be more time and more resource intense in a world affected by COVID. And so we've been doing virtual research studies now since 2012. First one we did was with 23andMe, and this is back when 23andMe was really a small company, and they wanted to confirm that people with self-reported Parkinson's disease, people who said they had Parkinson's disease, indeed did have Parkinson's disease. And we connected to those research participants in 50 different research participants in 23 states from a single site, and we did it in 3 months and confirmed that these individuals indeed did have Parkinson's disease, vast majority. Another study we're doing with the Michael J. Fox Foundation, funded by NIH, is we're following over 200 research participants with Parkinson's disease, I think in about 30 different states throughout the country. These are people who've completed a phase 3 study, so you can imagine this is almost long-term follow-up of these participants. In addition to getting an annual virtual research visit, just like we're doing right now, they are being followed with a smartphone application and an internet platform called Fox Insight to enable capturing of patient-reported outcomes. So here we're doing an entirely observational study of 200 and some participants across the country, single site, one IRB, and again, connecting to people directly in their home environment. And then I alluded to this in my opening comments in the last panel, as we're working with 23andMe and following LRRK2 carriers. We're doing virtual visits annually that include questionnaires, cognitive testing, including the Montreal Cognitive Assessment, remote examination, including completing the elements of the Parkinson's disease rating scale, except for a couple that can't be done remotely. Basically, all the same assessments that you would do in a clinical trial, we're doing in a observational study, and we're doing it entirely remotely. Again, following 250, about 300 individuals who are LRRK2 carriers, the vast majority of them without Parkinson's disease. And this is data from the first 3 months in which we enrolled over 100 participants from 28 states. Actually, in 3 months, we became the largest study of LRRK2 participants with the broadest geographic reach of any study ever conducted in this population. And we did it at a fraction of the cost of larger studies, such as those sponsored by the Michael J. Fox Foundation. So in 3 months, we had greater geographical reach for LRRK2 carriers than any other previous study, which had been almost exclusively or largely focused, for example, in New York City. So that's what we can do in terms of decentralized studies or virtual visits. And now I'm going to switch to digital biomarkers. So I think what digital biomarkers really do is help us see what we can't see in clinic. So in clinic, we only capture 0.1% of somebody's life. And what we do when they're in the clinic is we have them fill out various rating scales, like the rating scales I showed. Maybe we do a timed up and go test to get an objective measure of gait function. but largely we miss the 99.9% of the time that they're not in clinic. And I think we miss what's important. So for Alzheimer's disease, we miss the number of words that somebody speaks per day. In Huntington's disease, we miss what portion of the day that they're sedentary. And we found in one study that ambulatory individuals with Huntington's disease, a rare genetic disorder, spent half their day lying down. If we get sick with the flu, we might lie down half our day, but individuals with Huntington's disease are lying down half their day, and none of my colleagues or I had any clue that that was the case. For depression, do we know how much time people are spending alone or how much time they're spending outside the home? We don't even know some basic things in Parkinson's disease, like what portion of the day that people have tremor. Is it 3 hours a day? Is it 5 hours a day? What's the frequency of the tremor at home? What's the amplitude of the tremor at home? When does it occur? We have very, very, very limited insights into all these things, which are, critical to assessing whether a new drug, device, or intervention works or doesn't work. And neurological disorders or neuropsychiatric disorders are really, really well suited for digital biomarkers for two reasons. One, the quality of the current measures is low. Assessing people with Parkinson's based on an episodic assessment of somebody's finger tapping doesn't appear to be as good as, for example, having someone tap and getting objective measurements of their finger tapping in their natural environment, to be able to do so frequently, to be able to do so objectively, and to have very sensitive measures. Similarly, these digital biomarkers are well suited to measuring many key aspects. So you can measure voice, you can measure gait, you can measure, in multiple sclerosis, you could probably measure vision, lots of things that are affected. In pain, our colleagues at Evidation have demonstrated that people with chronic pain walk 25% less than people without chronic pain. And I would submit that might be more meaningful than asking someone after they've driven an hour to a research site in a car to circle a happy face that tells you how much pain that they're in. And these sensors, as you guys all know, can give you objective measurements. I think that's the Roche smartphone app there. Sensitive measures, frequent, and to do so in people's natural environment, and to do so remotely. And so we've done work with smartphones, wearable sensors from MC10, a radio wave sensor from Emerald, our colleagues at MIT, Dr. Dina Katabi, and video analytics, all to assess various neurological diseases. As Christian alluded to, our Center for Health and Technology works with pharmaceutical companies and is delighted to work with any of you on these approaches and conducting large-scale research studies or pilot studies. And these are data from work with Dr. Dina Katabi, a very talented computer scientist at MIT, and her radio wave device goes in somebody's homes and emits radio waves, kind of like sonar. Instead of sound waves, they're radio waves. They reflect off things that have water in them. And you can tell where a person is in their home environment. You can tell how fast they're moving, tell how frequently they go to the bathroom. You can tell how often they're at home, how many people are in the room. And this very first pass from like 2 years ago, we just wanted to ask how much time that someone's in bed. And so let's just look at participant number 4. And participant 4, each concentric circle is 1 day, and we observe people for 8 weeks. So we have 56 concentric circles, and starting at 0 is midnight, and then to the right is 6 a.m., 12 noon, and then 6 p.m. And you can see that this participant pretty regularly goes to bed at 10:30 at night and wakes up at about 4 or 5 in the morning. And then their average duration in bed is about 5.3 hours. Which is, short. Participant 2 is even shorter, 4.4 hours, but a regular pattern. Participant 7, again, a regular pattern. Hopefully that looks like something like our sleep behavior. But you can see participants 5 and 6 have completely lost their circadian pattern of sleep. And participant 5, you would really have a hard time as to when they're going to sleep. In this case, this study, we just said when they're in bed. We don't know if they're in bed because they're depressed or they're immobile, but that's what we're seeing. And then next, let me see. Next is our, Christian alluded to, we have a digital biomarker journal. It's open access. It's currently free to submit, and it's now indexed in PubMed, thanks to Christian and many of his colleagues who are on our editorial board. We'd love you to submit your digital biomarker work to us. And then our Center for Health and Technology is delighted to collaborate with you and your colleagues on neurological studies, because we think we'd love to help you develop therapies for the world's leading cause of disabilities. I'll stop there.
Thank you, Ray. Wow, that was a wild ride through the totality of decentralized trials and kind of a focus on digital biomarkers. Just want to remind colleagues on the call, submit your questions that you have to the POLEF, the link you find in the chat. There was a first couple of questions, and let me maybe start with one that is not digital biomarker specific, but really just go straight to the disease. And kind of, Ray, you mentioned that there is such a high number of Parkinson's patients around. And there's the question, is there any particular reason for this dramatic increase recently?
So obviously aging has a big factor, but we think Parkinson's is a preventable disease and is largely man-made and is tied to the use of industrial products and byproducts, especially certain pesticides. There's a pesticide called paraquat in the United States that increases the risk of Parkinson's by about 200%. There's a chemical called trichloroethylene that's widely used in degreasing, used in transportation and refrigeration. Contaminates half of Superfund sites in the United States, was banned in Europe about 10 years ago, and is contributing to the rise of Parkinson's disease. We have a whole book called Ending Parkinson's Disease. I'll see if I can pull it up for you right now. And it goes into great detail about this, and it even talks about the Roche smartphone app and some other things. I'll pull it up right here. Christian will let me do a quick plug for our book.
Absolutely.
If you want to learn more about Parkinson's disease and the treatments for it, we wrote it with Dr. Todd Scher, who's the CEO of the Michael J. Fox Foundation, and two global Parkinson's experts, Michael Oken and Boss Bloom, who might be familiar to many of you, and you can just order it on Amazon right now. It's been well reviewed, and it's written for the general public, so it's easy to read, a non-technical way to look at Parkinson's disease and why it's the fastest growing neurological disorder in the world.
Thanks, Ray. Definitely suggest that people who are interested in Parkinson's kind of get more insights from your book there. Another question to follow the sequence was around decentralized trials, and in particular, how kind of, our trials very often in the pharma industry are global trials, and how to break down the local ethic requirements and address those.
Local IRB. So most of our work has been done in the U.S., so there's lots of work to be done. My guess is that for decentralized studies that are global is that you're going to have regional or country center hubs that facilitate remote assessments in that country. Obviously, language is a barrier for some environments, and then there are different regulatory standards for them. We've had relatively little trouble doing this in the US. The EU obviously has different regulatory standards, but I think you're not going to be able to do research studies except for a decentralized approach. In the midst of the pandemic, I saw someone with Parkinson's disease in Italy because he was stuck in his home and he couldn't go see the doctor. And he reached out to me, by email. So I think for some of these things, the regulatory authorities and the IRBs and ethics boards, there is no alternative. Or the other alternative is, to expose people to huge risks. And I think the first research participants who gets hospitalized with COVID-19 will cast a large shadow on that study and all research at that site.
Very good. Now people are waking up, but unfortunately we have to move on already to the next call. So please, next time, enter your many questions earlier on, so we can bring them forward. Great. Well, thank you, Ray, so much. Excellent explanations, and looking very much forward to continue this collaboration with you there on very many fields, and see this all published in the Digital Biomarker Journal with Carter.
Thank you very much, Christian, and thank you to all your colleagues. Have a great conference.